Hello everyone, my name is Visalhounuo. In this video, I'll be explaining about slide culture technique. This slide culture technique, it is a rapid method of preparing fungal colonies for examination and identification of fungi. Fungi are identified mostly by close examination of its morphology and its characteristic it possesses. In slide cultures, the fungi are grown directly on the slide on a thin film of agar. Identification of fungi depends largely on their microscopic features like colony characteristic, growth rate, color, texture, diffusible pigments, exudates, aerial and submerged hyphae. And in case of microscopic features, it includes arrangement of spores and sporine bodies, arrangement of conidiophores, and the way in which spores are produced help in the accurate identification of filamentous fungi. This can be achieved by tease mount, a technique in which a part of growth is teased with needles, a drop of LPCB is added, covered with a cover slip, and then observed under the microscope. Although the most common technique used in mycology, identification is often difficult by tease mount method because of the dislodgement of conidia and spores from the conidiogenous cell. To overcome this, slide cultures are put up, which is considered best for preserving and observing the actual structures of fungus. The method was developed by Riddle in 1950 and currently several modifications are in use. The principle of this test is fungi are inoculated in small blocks of nutrition-deficient agar, medium like sebrodextrose agar or potododextrose agar, covered with a cover slip and incubated. After incubation, the cover slip is removed from the agar block and placed on another glass slide to which a dye such as lactophenylcotton blue may be added and observed for microscopic structures. Identification is made by microscopically examining the undisturbed sporulating structures as they were arranged during growth on the agar block under the cover slip. Fungi, when grown in nutrition-deficient medium, develop spores rapidly and adhere to the surface of the cover slip. The requirements are sterile Petri plates with a bent glass rod and glass slide. All these are to be sterilized. Then you will be needing a culture media for fungal growth, example is sebrodextrose agar. Then a spirit lamp, cover slip, inoculating band wire, filter paper, and fungal specimen or fungal growth. Coming to the procedure, aseptically with a pair of forceps, place a sheet of sterile filter paper into a petri dish. Place a sterile U-shaped glass rod on the filter paper inside the petri dish, and this bent glass rod you can sterilize it by flaming on the spirit lamp. Pour enough sterile water or distilled water on the filter paper so that it get enough moistened. And then on the U-shaped glass rod, place a sterilized glass slide. Then take the culture media of sebrodextrose agar. Using a sterile scalpel blade, cut a small block of about 1 centimeter square, and so that the small block should be in such a way that it will fit the cover slip. And then the cut block, you place it on the sterile glass slide inside a petri dish. And then using a sterilized bent inoculating wire, you can sterilize the inoculating bent wire by flaming on the spirit lamp. Then you take this bent wire and touch the fungal colony growth and then inoculate it on all the 4 sides of the agar block which you have taken and place it on the glass slide. After you inoculate all the 4 sides of the agar block, Place a sterilized cover slip on top of it and then cover the lid of the petri dish and then observe it for about under the microscope. To observe it under the microscope, first take another clean glass slide, place one drop of this LPCB, open the petri dish and then of your slide culture and remove the cover slip and place it on top of the drop of the LPCB mount which you have taken and then observe it under the microscope. Precautions. Slide culture of slow-grain organisms suspected to be dimorphic pathogens such as Histoplasma capsulatum, Blastomycosis dermatitidis, Coccidioides immitis, Paracoccidioides brasiliensis should not be made. Slide cultures must be observed only after a cover slip has been removed from the agar plug and not while it is positioned on top of the agar plug. The latter method of observation could be very dangerous as it could cause a laboratory-acquired infection. Advantage of this slide culture are: first is, it is a cost-effective, rapid method for the identification of fungal isolates based on their morphology and microscopic characteristic. Second is, as fungi grow directly on the slide on a thin film of agar, there is no need to remove a portion of the fungus from a culture plate and transfer it to the slide. This reduces the chance of damage to fragile reproductive structures or spore-bearing structures of fungi. Disadvantages of these slide cultures are: as the agar medium used contains a limited nutrition source, fungal pathogens will survive for a short time period. Therefore, this method is not suited for fungi which take more than a few days to reach maturity. For example, like the members of Scilaricia and many fungi isolated as endophytes.